In this question, you have to identify which one is least likely to be involved in stabilizing the three-dimensional folding of most proteins. Option A, hydrophobic interaction. Now, hydrophobic interactions are important in stabilizing the three-dimensional structure of the proteins because non-polar side chains tend to cluster together in the interior of the protein to avoid contact with the water, thus contributing to the protein stability. Hence, it is incorrect option. Option B, ester bonds. Now, ester bonds are usually found in lipids such as triglycerides and not in protein structure. Protein structures are primarily stabilized by other interactions such as hydrogen bonds, hydrophobic interactions, and electrostatic interactions. Hence, this is the least likely factor and thus it is the correct option. Option C, hydrogen bonds. Now, hydrogen bonds also play an important role in stabilizing the secondary, tertiary, and quaternary structures of proteins. Hence, it is incorrect option. Option D, electrostatic interaction. Now, electrostatic interactions or ionic bonds are very important in contributing to the stability of the protein structure. Hence, it is incorrect option.